Yeah, so thank you everyone for joining today. For those of you who don't know me yet, I'm Alex from the Molecular and Genomics Informatics core. We also have today Pradeep Kumar, who's a lab host in Echo, and then also Carol Ann, who is from the Echo team. And she's going to tell us a whole lot more about this awesome machine. But before I turn it over, just general housekeeping items. If you have any questions, please drop them into the chat or the question and answer box. We'll monitor and kind of do what we can on the fly. But anything that's not mentioned during the talk, we'll get to at the end. So with that, I'll turn it over to Carol Ann, who's going to tell us more about the Echo.
Thanks so much, Alex. Thanks for having me today. Hi, everyone. My name is Carol Ann Radaman. I am the Automated Solutions Consultant for Beckman Coulter Automation product line. What that means is that I have conversations with customers about their workflow, and we try to decide if Beckman Coulter has a technology that can help them to automate some of their current processes. If anyone has a question, please, like Alex said, drop it into the chat. Alex, I'll probably stop at points during the presentation to ask if anyone has questions, so we can just go over them then. And if there's any questions that people have today that I don't know the answer to, I will be sure to find it out from people who are much smarter than I am and forward the answers along to Alex. All right, my slides don't seem to be moving. There we go. Okay, so just to briefly go over the agenda, we're going to talk about our entire life sciences portfolio, then specifically drill into the liquid handling portfolio, why we have this Better Together promotion, which is tip-based liquid handling and acoustic-based liquid handling together, and then go over some Echo technology and some specific case studies that have been done on the technology and why you should think about incorporating it into your processes. So as it stands today, our automation portfolio has two distinct product lines. The first one is the tip-based liquid handler product line that you could see along the top here. The Biomech 4000 was actually obsoleted last year, so ignore that one. But we have the Biomech, the i5, and then the i7. The i5 has one pipetting pod, the i7 has two. Then we have larger integrated Biomech workstations, and then we have much larger integrated solutions that often involve one robotic arm or several robotic arms, depending on what you're trying to do. So moving from the simplest setup from on the left-hand side all the way through the most complicated to the right. And then the same for our Echo acoustic liquid handlers. So we have our lower throughput options on the left-hand side with the Echo 525, then the Echo 600 series, then access workstations that can incorporate up to 6 integrated devices, and then our single robotic and dual robotic systems that are all Echo-centric. So no matter what you're doing in your lab, we have scalable solutions for your liquid handling workflows, both at a microliter scale for the tip-based liquid handling and then at a nanoliter scale for the acoustic-based liquid handling. So what we want to do is have a large variety of liquid handlers to support all of your applications. So whether you're falling under the genomics umbrella or the drug discovery umbrella, we have something that can fit that, and we can have a conversation about what that looks like. If it's a hybrid approach of acoustic-based liquid handling and tip-based liquid handling, that's great. We have something that can do that. If it's just one or the other, we can also have a conversation about that. And that's specifically designed for each customer. So we don't have kind of off-the-shelf solutions for you. We really want to have an in-depth conversation about what you're doing and decide which portfolio and which instrument is best suited for that. And then later on, if you decide that you want to scale up or move into a higher throughput part of the workflow, then we can absolutely help you with that as well. And it's all scalable and modular, so you can start small and then add on later. In addition to myself, we also have a team of field application scientists and field project managers that get involved when we talk about those larger automated opportunities with robotic arms and multiple devices, just to make sure that we have a smooth implementation and you're well supported on the application side of it. So now let's drill into the acoustic-based liquid handling portfolio, starting with the Echo 525 on the left-hand side. This Echo is designed to transfer mostly aqueous fluid, so we can transfer small percentages of DMSO, but mostly we're in that aqueous category. So this has a lot of uses in genomic applications, synthetic biology, and proteomic workflows. The ECHO 525 transfers 25 nanoliter droplets, and we'll talk a little bit more about that technology when we get further into the presentation. And you also have the option to transfer from an ECHO qualified reservoir, which is a 6-well reservoir that you can put into the ECHO, and that has a lot of uses when you're transferring primers or master mixes for your genomic and synthetic biology workflows. Then just moving right on the screen, we have the ECHO 650. This is ideal for medium to high throughput workflows. The 6 series transfers 2.5 nanoliter droplets, and the 6 series ECHO also has the ability to accommodate 1536 well plates. So again, just moving up in that throughput. This is more for DMSO based liquids. It can do aqueous as well. And then we also have the acoustic tube upgrade available, and that has a lot of uses in drug discovery workflows and small molecule workflows. And again, just keep moving right on the screen. Then we have the ECHO 655. The only difference between the ECHO 650 and the ECHO 655 is the speed of the motor. So the ECHO 655 is about twice as fast as the ECHO 650. So this is really for when we're moving way more into that high throughput category. Then still moving right on the screen, we have our ECHO automation. So it's compact, it's modular, it's flexible, it's scalable. Each one is designed specifically for whatever your application is. So a lot of the times there aren't access systems or single robotic systems or dual robotic systems that look anything alike. And that's because we want it specifically to be tailored to your needs. So we have a software that's a scheduling software that controls these larger automated systems. It's pretty easy to use. I'm not a software person and I can wrap my head around it. So if I say that, then it must be pretty easy. We also have a full portfolio of ECHO applications that we're going to talk a little bit about as well, and it integrates with the scheduling software. And when we design these automated solutions, we make the protocols echo-centric, so your ECHO is as busy as possible. We want to constantly be feeding those plates. So once you're proficient with your ECHO protocols and you feel like you're running that 30, 40, 50 plates a day, and you're just kind of staying there all day feeding the ECHO, that's really when we want to talk about these automated systems, because instead of you standing there and feeding the ECHO, we can have the robotic arm do that for you instead. So that's really when we start having those conversations. Now, just briefly, we're going to talk about some of our NGS library prep solutions, and this one is specifically focused on NGS sample prep. And what we've done is we've actually integrated an ECHO into a Biomech liquid handler. So the ECHO 525 will take care of the small volume transfers that are needed and the parts of your protocol that you would want to save reagents with. So when we're talking about those expensive primers and the expensive master mixes, that's what we want to use the ECHO for. So we integrated the ECHO 525 with a Biomech i5, which has that single pipetting pod. And this Biomech i5 has a 384 multi-channeled head. And the i5 is used for when we need to do full plate additions or and dispenses or aspirations. And then it's also used for the bead-based cleanup portion of the NGS sample prep. And the bead-based cleanup, that's one of the limitations of the ECHO, is that we can't transfer beads because they're just too heavy. So once we get into the technology part of the ECHO, we'll talk a little bit more about that. But I just have a short video here that shows how the ECHO is integrated directly into the Biomech. And you could see back here, there's an automated thermal cycler as well. And there are some heating and cooling and shaking positions that are on this to complete the NGS workflow. So right now, the gripper is just grabbing the plate that is the source plate for the echo, and it's going to pull the plate directly into the echo, and then the gripper is going to go and grab the destination plate. And what's interesting about the destination plate is that when it's placed into the echo, it's actually flipped upside down. So customers always like to ask, well, how much volume can I flip upside down? And my answer is, that depends on a lot of things, including the surface tension of your destination plate and also the viscosity of your liquid. So try it. Flip the plate upside down on the lab bench. And oftentimes customers will find out exactly where that happy medium is for them. So then the echo is going to perform the senses, and then it would eject the plates back into the biomech to complete the workflow. Just a cool piece of technology that we use and combine our portfolios. Does anyone have any questions at this point?
I think we're good so far.
Okay, great.
Thanks.
So now we're going to specifically talk about the acoustic-based liquid handling. So why should everyone invest in an echo in their lab? There are many, many reasons. First of all, we're going to minimize your costs. And the echo up front is an expensive piece of equipment, but we have done some studies, actually many studies, on where your savings are. One of those savings is going to be on tips. We have plates that are specific echo qualified plates that are the source plate. And again, we'll talk about why that's important in a few minutes. But you can seriously reduce the tips that you use in your lab. And by doing that, you're also helping out the environment because you're not creating so much plastic waste. The other thing is that we conserve sample and reagents when you use the echo, because our ultimate goal when you purchase an echo is to minimize your reactions as much as possible and still help you get the same results. And again, I'll show you some studies where we've done that with major pharma companies that have published with us, and we can prove that we can still get the same results. The other thing we can do is really reduce workflow steps, and by doing that, we can save you time, and time is money. So we want to make sure that we are giving you that ROI up front. Another reason is we can increase your data quality, so we can minimize the number of reruns that are necessary because there's less error propagation. Also, reducing your assays to nanoliter scales allows for additional flexibility, and if you do need to rerun a plate, it's not nearly as expensive. We can also do dispensing directly from stock solutions, which is key in drug discovery workflows. We can accelerate your productivity. You can change, design, test, and optimize all your assays very quickly. The software, again, is very easy to use, so you can change it as needed on the fly. You can also transfer multiple fluid types and concentrations from a single plate, so you can preload that plate the way that you need to run it, design your workflow within the software, and it's color-coded, so it's easy to see. And then you can just tell the echo exactly where those fluids need to go. We can automate your complex assays. We can transfer low CVs and with high accuracy. And we can have a large dynamic transfer range. So even though we're really talking about nanoliter at this time, we can also do low microliter. And again, the limitation on that is the surface tension of the destination plate and the viscosity of your liquids. If we can flip it upside down, then we can transfer to it. So as long as there's no reagent leaking out of the destination plate, then you're golden. The other thing is that the echo will print out a transfer report for every single transfer that you do, and it will let you know if there are any errors or if there wasn't enough volume in the well, etc. So you can easily keep track of that. We can also enable compliance through our software with something called Echo Compliance Manager. And then we can define it further using user permissions management. So you can track who went into your protocols, made any changes, things like that. So we make it easy for you to track it. And if you were subjected to an audit, we can help support that process as well. So we have a full portfolio around the Echo, first with the automated solution. So again, that's for your scaled up processes with the robotic arms, so you don't become a human plate feeder for the Echo. Then we also have consumables. So our acoustic qualified source plates and tubes are manufactured to a very tight CV, and you'll see that in the next video. And the reason for that is because the transducer that performs the acoustic droplet ejection needs to understand and be able to calculate the flatness of the bottom of that plate. If it can't properly do that, then it won't properly transfer the fluids that you've designed. Then we also have a full complement of software. So when you purchase an Echo, we talk about your protocols and workflows that you'd like to do. And then we assign the software packages a la carte. And the reason we do that is because the software packages are expensive. And a lot of the times you only need 2, maybe 3. You don't quite need all of them. The most common ones that my customers use are Cherry Pick, Echo Cherry Pick, which is right here, and Echo Plate Reformat, which is right here. And then I would say a close third is Echo Dose Response. Other than that, Array Maker, Plate Audit, Combination Screen, you won't see all that much. I would say mostly Cherry Pick and Plate Reformat can get that done. But obviously, if you're looking to do any dose response curves, then you need the dose response software. Any questions, Alex?
No, I don't see any in the chat yet.
Great. Okay, so one more time, again, just moving from left to right, we're talking about scaling up. So Access workstations can be integrated with up to 6 devices, and it can go right on the lab bench. The single robotic system is a standalone system, so it is scalable and modular, just like our other systems. We also have environmental options, so if you needed that, we can accommodate that. And then our dual robotic system is for 12 plus devices. So again, same options. It's scalable, it's modular. We have the environmental options. The other thing that we are capable of doing is actually integrating with Titian and LIMS, and we can help support that. We have dedicated automation and application service teams to make sure that you are up and running as soon as possible and as smoothly as possible. So this is our Echo video that I was talking about before. So I'm going to just talk my way through it, and then we'll discuss what parts were really important. So the source plate is being placed on the source stage within the Echo, and then it's going to pull in. The destination plate is placed on the destination stage, pulled in and flipped upside down. Then the transducer is going to perform what we call a survey, which it's going to visit each well in the plate. It's going to fire three pulses of sound. The first is to get the plate bottom reflection, then it's to get the well bottom reflection, and then it's going to detect where the meniscus reflection is. So it's going to do that for every single well in the plate. It's going to go back to the first well that you've designated in your transfer, and it will know exactly how much sound energy it needs to fire a droplet of liquid from the meniscus. And what it will do, if it's a 525, is fire a 25 nanoliter droplet. If it's a 6 series, it will do a 2.5 nanoliter droplet. So whatever your protocol has laid out, it will calculate the number of droplets it needs to meet that volume transfer request, and it will do that from each well. So you could see the destination plate is the one that moves. The source plate stays completely still, and the transducer just moves underneath the plate. Customers always ask me how fast is the Echo, and I don't really have a great answer for that. All I know is that it's pretty much the fastest thing I've ever seen. It takes longer for the plates to be pulled into the Echo, which you saw in the earlier video, than it does to actually perform the transfer. So it says 700 droplets per second, and it's really not kidding. I don't think I've seen a plate go in the Echo for longer than 2 minutes, which is way faster than any tip-based liquid transfer I've ever seen. Earlier I mentioned that the plate is manufactured to a very tight CV flatness on the bottom, and that's because that transducer needs to travel. It has the coupling fluid that's over the transducer itself, and the purpose of that is to enable it to detect. It's like when you go and get an ultrasound at the doctor and they need that gel to actually be able to see anything. It's the same idea. So when that transducer is traveling underneath, it's carrying that coupling fluid with it. If you try and use a not an echo qualified source plate, the echo will error out. It won't let you proceed, and that's because it won't transfer properly. So our precision, our published specs are always more conservative than what the echo is actually capable of, but it's less than 8% of precision, and accuracy error is less than 10%. So it's very accurate, it's very precise. That's not usually the service issues that we have with the echo. So again, we're trying to reduce volume and therefore reduce costs. We want to improve your accuracy and precision of your assays, and we also have very, very few cases of cross-contamination. And that's because there's nothing touches the liquid. The only thing that the liquid is subjected to is the sound energy. So now we're going to talk about different applications that the echo has been very important in. So in drug discovery, as everyone knows, drug discovery assays can change at a moment's notice. So through our software, we make it easy to change your protocols and also keep track of those changes that you've made. So we can also reduce your hazardous waste. We can reduce your tip and plate waste that you create when you're running your assays. We can preserve your library and sample integrity, and the way that we do that is by transferring directly from your stock solutions. So a lot of customers have their stocks in these acoustic tube racks, which we are validated on, and those are sold through Azenta, and we can transfer directly from those acoustic tubes. So the 6 series was acoustic tube upgradable, and we can absolutely help preserve those stocks. Then when we can create the intermediate dilution plates as needed and then go right to your assay ready plates, or we can go directly to your assay ready plates depending on your workflow. And these are easily reproducible because we have everything recorded in the software. So one thing that the echo is really popular for is direct dilution. And when you perform serial dilutions on a tip-based liquid handler with a manual pipette, you're transferring the same volume in decreasing concentrations. And this can result in error propagation throughout your assay. So if you make one mistake, then the whole assay is going to be subjected to those errors. With direct dilution through the echo, we can transfer the same concentration in decreasing volume. So because the echo has the nanoliter capability, when we do a dilution plate, we just transfer from the same solution and just decrease the volume for each consecutive well. This means that we can reduce the number of dilution steps necessary and still maintain that precision. There's no carryover or binding because it's touchless transfer, which means you have more accurate potency values. So using acoustic transfers, we have worked with all of the top 25 pharmaceutical companies, and that's worldwide. So this left side here was a study done by Bristol Myers Squibb, actually, and they published this. So what this is showing is that using tip-based and using echo-based dilution, they evaluated 1,100 compounds, and 46% additional compounds were found to be potent that were missed by traditional tip-based liquid handling. So 68% were found to be potent by both methods, and then the echo transfer identified only identified an additional 32%. So we can still maintain your sample integrity and find more hits. So then AstraZeneca did this study on the right-hand side. So they discovered an entire class of cancer therapeutics by using the echo liquid handlers. And it was missed when they performed the same assay with tip-based liquid handlers. So we can make your assays more potent and compounds over 200 times more potent through the echo transfers. So they published that, and I can always send over the white papers on those. In combinatorial screening, we can track your concentration quality of sample to ensure it's reproducible. So this is a screenshot from our software. I really like these screenshots because it shows how easy it is and the plates are color coded so you can keep track of everything. So you can see that these are pretty complicated assays, but within our software, it's easy to map out exactly where everything is going because we keep track of it with those colors. We can also decrease the effects of DMSO because we have such low dispense volumes, and we can reduce the number of cells that are needed in your plates. In addition, you can see from here that we can perform any well to any well dispensing. So you can optimize your plate layout and we can minimize your variation. Then we can perform different drug combinations because of that any well to any well capability. And then just to switch back over to some genomic applications. So RT-qPCR is one of our most common uses for the echo portfolio. And most of that is because we want to preserve, we want to save on the master mixes and primers because they're expensive. So we can use our Biomech or tip-based liquid handler to perform the initial RNA isolation and purification. We can also use the tip-based liquid handler to set up the source plate. So you can transfer your DNA samples, you can prep one plate with primers, probes, and some companies like IDT have commercially pre-filled options that come with the kits. And then we can also prep your master mix reservoir. So then we can prep your PCR plate in the echo. So you can put your DNA sample plate in there, then you can put your primer plate, and then your master mix plate. And then we can take the echo plate, the destination plate out, and it can go directly into a PCR machine. So because of those low volume additions, we're saving money and you're also saving time. And we can easily reproduce them. And you can see from here, we perform these studies on a 20 microliter reaction versus a 2 microliter reaction, and you can see that the results are very, very similar. So even though we significantly reduce the reaction volume to save money and time, we still were able to have the same results. So we can save reagents, save time, and enable your workflows. Then we also did some work with the SMART-Seq single-cell RNA library prep. So first we use the echo to add the lysis buffer to the plates, and then we sort the cells using a cell sorter. Then within the echo, we can add primers, add the master mix, and then add the second strand synthesis mixture. And then we can go to a regular tip-based liquid handler for the cleanup because, again, we can't do beads or columns in the echo. And then it can go directly to library prep. And this saves a lot of time for your NGS library prep workflows. So again, here are some results. So we did it at different miniaturization levels. So you could see we still get very similar concentrations and still have sufficient cDNA across the input no matter how much we miniaturized. There was no significant difference in expressed genes through miniaturization. So we're still able to save money and save time even though we miniaturized your assays so much. So lastly, we have a full complement of workflows that can be incorporated into your lab. We have instruments that are designed specifically for what your workflows are. We have software that enables you to focus on your workflows because they are user-friendly. We are constantly expanding our applications portfolios and working with large kit manufacturers and large pharma to drill into their drug discovery workflows or genomic workflows to expand what we can do. One of the coolest things about working with the Echo is that a lot of the times customers will say to me, can we do this with the Echo? And I say, well, I don't know. No one's ever done it. Let's try it. And then we find out it can or it can't. And that's pretty cool to have customers always working with us to expand what we're capable of. Then also to complement the applications and the hardware and the software, we also have consumables. So we have genomics kits as well as tips and plates to go on the instruments. Pradeep wanted me to make sure that this was included. They have a Echo 650 acoustic liquid handler, and it's located at this address, and this is his contact information. And that's it for me. So, Alex and Pradeep, I'll pass it back to you.
All right. Thanks for a great presentation. Yeah, Pradeep, did you have any additional words you wanted to add?
Yeah, I just want to make sure that people understand that we have this available at Public Health Research Institute. And our lab has been using this instrument for many years. We had an instrument before, and now this is a new instrument we got through the NIS shared equipment grant. And we have been able to produce very consistent results, especially in drug discovery field, where we can do multiple drugs synergy assays within plates in like no time. Basically saves a lot of time, hands-on time, as well as the reproducibility of the system is really great. So all Rutgers wide, the instrument is available for any users to try or use for large scale library reformatting of their large scale libraries they have into assay formats, or just making plates, MIC plates, or at large scale for doing any type of assays. Instrument can do up to 1536 well plates, starting from 96 well to all the way up to those plates. And we are happy to help. Reach out to me. The instrument information is also available at the PHRI website if you go into equipment and services section. So we are happy to help any researcher at the institute who may want to try it or use it for their research.
Awesome, thank you.
And I want to thank Carol Lian as well for giving this presentation today. It was really awesome to have you back speak.
Well, thanks for having me. I appreciate it. And if anybody has any questions, I'm happy to take them. So please pass them my contact information. I'm always happy to have a conversation.
Right. Perfect. Well, thank you very much, everybody. And yeah, any questions we'll forward along. And I guess until next time. So thank you.
Okay. Thanks so much. Have a good one.
You too. Thanks.
Okay. Bye.